Welcome to the International Stem Rust Screening Site here at Njoro. KALO stands for Kenyan Agricultural Livestock Research Organization, and this screening facility is supported by Durable Rust Resistance in the Wheat Project. So we've got about 13 hectares of quarantine facilities to facilitate testing of international germplasm here at this site at Njoro. So Robin is one of the varieties that was released in 2009. This variety has become quite popular with farmers until last year that we have started to see these huge susceptible pustules occurring on the stem. This variety has high yield, better than the local varieties, but at the same time it is based on a single major gene called SRTMP and some APR genes in the background. It has been resistant for almost 5 years when this variety was deployed. Interestingly, from last year we have been observing these huge stem rust pustules, which is indicative of the fact that the gene is no longer effective. This could be of the fact that the new race that has been reported in Ethiopia conferring susceptibility to the same gene is also prevalent in Kenya, or it could also be of the fact that there could be another race which is of the same UG99 lineage with added virulence to SRTMP gene, which needs further confirmation. So this is a typical example of a boom and bust cycle where an introduction of a variety which carries single major gene has become a mega variety in Kenya, almost occupying 30% of the area. But then of recent we have started seeing this new race which is rendering this variety susceptible, and most of the farmers who are growing this variety would get yield losses. On the other hand, having different varieties in place ensures the fact that there is immediate or rapid replacement of such susceptible varieties that are broken down with better resistant varieties with the different gene combinations in place. This is Kingbird actually. As you could see this line appears quite clean to the disease. Many of them would assume that it could be a major gene behind such kind of resistance that you are observing. But interestingly, this is line Kingbird. Kingbird is developed by CIMMYT, and this line does not carry any major genes. So this line is susceptible at seedling stage, but as it reaches the adult plant stage, you could see the expression of APR genes in the background, and this is a combination of 4 to 5 minor genes which has led to similar effects of a major gene, wherein you could see a clean phenotype. And on the other hand, this also has pseudo black chaff. As you could see, this is a purplish pigmentation that is associated with stem rust gene SR2, which is accumulation of melanin not only on the glooms and awns, but you would also see sometimes at the base of the internodes. And the importance of this gene, this is a race non-specific gene, has been effective for almost 90 years and has been widely used in CIMMYT breeding program. And it has been always a background of the CIMMYT breeding program for resistance to stem rust. And this started as early as Borlaug, when he had started off in CIMMYT to address the issue of stem rust, that he incorporated SR2 into several of the CIMMYT breeding materials. And one of the outcomes of such APR genes in combinations is this variety, which has been released in Kenya and has been consistently been resistant for almost about 5 years. The battle for rust resistance is always a continuous ongoing process, and the more you shift towards resistance, the pathogen also tends to survive and tries to mutate to produce new virulent races that tend to defeat the genes. So the better approaches are to have combination of genes, even though if one gene gets broken down, there are additional genes that can confer resistance to ensure that the farmers do not get losses or you don't see an event of epidemic occurring in the near future.